Hi, Dr. Cook and class. I'm Chelsea Seiler once again, and today I'll be talking about glycolysis, gluconeogenesis, and the role of ADAlase gene expression. So to start off with a topic summary, this presentation is going to be covering glycolysis and its mechanism, gluconeogenesis and its mechanism, aldolase, what it is, and its isoforms, the expression of aldolase in the body, and regulation of aldolase B. So let's start off by talking about glycolysis and what exactly that is. Another name for glycolysis is the Enden-Meyerhof pathway. I hope I pronounced that right. And it is the first step in cellular respiration. It's done anaerobically. Oxygen is unnecessary for glycolysis to take place, so it can be done by aerobic organisms and anaerobic organisms. Glucose, in essence, is broken down so that 4 ATP molecules are released, 2 NADH molecules are released, and 2 pyruvate molecules are released. And then that pyruvate is going to be used in the citric acid cycle, which will lead into the electron transport chain. But that only happens if aerobic respiration is taking place, because everything after glycolysis in cellular respiration requires oxygen to happen. If it proceeds anaerobically, then that's a whole different process. So taking a look over to the diagram on the right, we see a very, very abbreviated diagram of glycolysis. So we have glucose on the left, that hexagonal sugar, and as you go through the process of glycolysis, we see these 2 NAD plus molecules are reduced into 2 NADH molecules, and 2 ADP molecules are phosphorylated to become 2 ATP molecules overall. Now, I know that it says over here 4 ATP molecules, but you're only seeing 2 in this diagram. And the reason for that discrepancy is because it takes a little bit of ATP to make some ATP. So our net gain overall is 2, even though 4 are technically produced. And then the end result of glycolysis over on the right are these 2 pyruvates, which if you count the amounts of carbons there, there's 3 in one molecule, 3 in the other, which makes sense given the law of conservation of mass, given that we started with a 6-carbon glucose over on the left. So taking a look at this slide, this is the mechanism of glycolysis. Glycolysis consists of 3 overall phases and 10 specific steps, which you can see numbered here all the way down to the bottom. This entire diagram kind of travels in a zigzag pattern. So let's go ahead and zoom in and look at the individual phases and steps of the glycolysis pathway. So this slide consists of glycolysis phase 1, where glucose is trapped and destabilized. Now, if I were teaching glycolysis to, say, a high school biology class that wasn't an AP class, I might just leave it at that, leave my explanation at glucose is trapped and destabilized in phase 1. But the more advanced the class, the more specific you should look at the exact mechanism. So over on the left over here, I have the descriptions of what specifically happens in each step. And on the right in this diagram, on the far right, these over here, we start with glucose, and at the end of each step, you'll see the results of what that carbon-based molecule is going to look like. And then this series over here is kind of an abbreviation of what's going on from this description over there. So looking at step 1, hexokinase is going to phosphorylate glucose, yielding glucose 6-phosphate. So let's take a look at what that looks like. Up on the top here, we have the original glucose molecule, again, that hexagonal sugar. Hexokinase is the enzyme that's going to catalyze this reaction using ATP. So it's going to take that phosphate off of ATP, making it ADP, and it's going to stick it onto that glucose. And so the result of that step we can see right here, and that phosphate got stuck on right at the top there. Now, as I stated before, this does use ATP. It requires ATP actually to perform this phosphorylation. And this phosphorylation is actually considered to be irreversible, meaning it would be very energetically unfavorable to go backwards in the same way. So you would need some energy input to go backwards. So that's step one. Now, looking at the result of step one, the glucose 6-phosphate, which again is this molecule right here with that phosphate stuck on up top, the next step is going to be the isomerization of G6P or glucose 6-phosphate into fructose 6-phosphate, which is this one right here. Now, the enzyme phosphoglucose isomerase is going to perform that, and we can see by this little double-pronged arrow here that this is a reversible step, an easily reversible step. So a couple of things are happening during this transfer between this step up here and this step down here. One thing that you'll notice is that we don't any longer have a 6-carbon ring. The ring itself is made up of 4 carbons and an oxygen, I should say 5-carbon and oxygen ring up here. But that additional carbon that was here is now getting pushed out of the ring. So this is the same carbon that was in the ring at the end of step one, but is now not part of the ring in the end of step two. And you'll notice that this group has scooched over here, but it is the exact same group. That didn't change at all. It just has changed orientation. And if you follow all of the OH groups, you'll notice that they're in the same places on the same carbons. It's just that they've shifted a little bit around. So our next step in step 3, we're going to go from this molecule right here, from fructose 6-phosphate, and phosphofructokinase is going to add a second phosphate on. Again, we're going to use ATP to do this by taking a phosphate from that ATP and sticking it on. And so that phosphate is going to be stuck on right there, and you can see it go on. This step is also irreversible, meaning it would take an enormous amount of energy to go backwards. And interestingly enough, this is also the rate limiting step as well. So to go ahead and summarize all 3 steps once again, we've seen a couple of things happen. We have added a phosphate on right there. We have pushed this carbon outside of the ring, and we have added on another phosphate over here. Moving on to discuss phase 2 of glycolysis, 2 3-carbon molecules are going to be created by the cleavage of that fructose. So in this phase, we're actually going to split that entire fructose ring now in half, and we're going to get 2 3-carbon molecules out of it. So in phase 2, step 4, that fructose ring that we saw at the end of phase 1 is going to get split into 2 molecules, which are either DHAP, dihydroxyacetone phosphate, or glyceraldehyde 3-phosphate, which is abbreviated G3P. And the enzyme aldolase, which I'll talk about more later, is going to be in control of that reaction. So looking at the diagram down here, the end of step 3 is cut off, but aldolase, that's this enzyme here, is going to create this molecule, that DHAP, and also this molecule right here. And then in step 5, these two molecules are actually in equilibrium with one another, so they kind of move back and forth a little bit. And triose phosphate isomerase is going to control those two molecules moving in equilibrium with one another. So moving on to phase 3 of glycolysis, this is where ATP and pyruvate are going to be ultimately generated. Our first step of phase 3, or step 6 overall, is going to involve that G3P getting transformed into something else, into 1,3-bisphosphoglycerate. So here, you can see over here that that's done through the reduction of NAD plus to NADH, and you end up with this molecule right here, which we can see has two different phosphoryl groups on it. And then in the next step, we're going to see phosphoglycerate kinase removing one of those phosphates, this one up here, and turning that molecule into a different one, into just 3-phosphoglycerates. Now, I would like to note that it's not just one of these 3 carbon molecules that's going to go through this process. From now until the end, it's going to be 2 total molecules going through this process as I describe it, which is why everything is plural over here. So just keep in mind that there's 2 3-carbon molecules present per molecule of glucose that started this glycolysis process. From that step, from step 7, where that secondary phosphate group is removed, you get some ATP here. You get some ADP turned into ATP. So here's where 2 of those ATPs come from. And then in step 8, we're going to see that 3-phosphoglycerates transforming into 2-phosphoglycerates through some enzymatic action there. And these 3 steps that I've just talked about, these are in fact reversible, and you can tell that by the equilibrium arrows that are present in these diagrams. So looking a little closer at that step as we move from this molecule to this molecule, you'll notice that that phosphate down here changes position a little bit. Instead of just being removed or something like that, the phosphate that is here gets moved up and is now in this position up here. And that reaction, that movement is controlled by phosphoglycerate mutase. Then in step 9, we're going to see enolase controlling a reaction that's going to turn those 2 phosphoglycerates that we got up here into a phosphoenolpyruvate. So ultimately, looking at what's going on, we see that the structures are written a little bit differently, but what we're really seeing is a double bond being added in right there. And to do that, we're losing a water. So it's a dehydration reaction. And so we're going to lose a hydroxide, that OH, and then an H from somewhere else. So ultimately, water is lost there. And then in our last step, in step 10, we see pyruvate kinase turning that PEP, that phosphoenolpyruvate that we got up here, into a regular old pyruvate. And we're going to get more ATPs forming as a result of that step. It is also going to be considered one of those irreversible steps. And so what we're seeing in the transformation structure-wise is we're seeing this phosphate go away from the phosphoenolpyruvate molecule because we don't see it over here. And then that phosphate is going to be transferred and placed to form ATP ultimately. And then after all this is over, that NADH that we formed way up here is going to be recycled back to NAD+, and that's going to be done through a process known as oxidative phosphorylation. So next I'd like to move on and discuss gluconeogenesis. So what exactly is gluconeogenesis? Well, it's the synthesis of glucose from sources that are not carbohydrates. A lot of times you'll see gluconeogenesis thought of as kind of the exact opposite of glycolysis, and in some ways it is. They do share a lot of the same steps in reverse, but it's not it's not just there to do the opposite of glycolysis. Really, humans don't eat only glucose, only sugars. Sometimes we need to derive energy from other sources, such as fats or proteins. So gluconeogenesis it exists to provide our body with a usable sugar that's derived from a different source. Because if we could only eat sugar to survive, we couldn't eat anything else, and a lot of our diet would be very restricted. So that's part of what it's there for. And some organs in our body can really only utilize glucose, or only mostly glucose as their source of chemical energy. So we need to be able to create it from a source that's not glucose for that reason. Examples of that would be our brain, our eyes, our kidneys, our liver, small intestine, and there's a few others as well. So taking a little bit closer of a look at the gluconeogenesis pathway, right next to glycolysis, you're going to see a lot of similarity between the steps in reverse. So gluconeogenesis here starts out at the bottom, whereas glycolysis is going to start out up there. And if you look closely, you'll see a lot of similarity, particularly in this middle section here, with about 7 out of 10 of the steps of gluconeogenesis being shared with glycolysis, but just again, in reverse. So all of those portions of glycolysis that were reversible are pretty much going to be the ones that are shared with gluconeogenesis. But to zoom in and talk a little bit more specifically, we're going to see carboxylation of pyruvate, again, where we're starting out for gluconeogenesis, and we're going to form oxaloacetate from that. That is going to require ATP, and it's catalyzed by pyruvate carboxylase. And then that oxaloacetate, is reduced to something called malate by NADH, which is oxidized using that NAD+, and then reduced back to oxaloacetate ultimately. Unfortunately, in this diagram, that particular step that I just summarized is not pictured here. This diagram is going to go straight to the next step, which is the phosphorylation of that oxaloacetate to this molecule up here, the phospholynyl pyruvate, by the PEPCK, or PEP carboxyl kinase. That enzyme is going to control that part right here. So we've landed ourselves at this step right there. And then from here to up until that fructose 1,6-biphosphate is converted to fructose 6-phosphate up in this region. The mechanism ultimately in the middle is very much the same as glycolysis, just in reverse. So up until you hit here, that is pretty much the same. And then from here, the fructose 1,6-biphosphatase is going to convert the fructose 1,6-biphosphate to fructose 6-phosphate. And then in the last part, glucose is going to be generated by hydrolyzing glucose 6-phosphate and releasing that organic phosphate. Here is where you see that happening on the diagram. And then the glucose from there, once it's been generated, is going to get transported to the cytoplasm, which again could be used then for glycolysis, for generating energy, or to balance blood glucose levels for any number of reasons. The cells in various places in our body might need to go through this process. So at this point in the lecture, were I presenting this to students, I would stop for a knowledge check, just to make sure that my students are still engaged, still paying attention. So my knowledge check is for them to fill in the missing terms to the appropriate boxes using this term list right here. And again, some terms may be used twice, and it's their job to figure out where everything goes. So next, I'd like to talk about aldolase, a particular enzyme that's also known as fructose biphosphate aldolase, Looking at this ribbon diagram on the right, we can notice a couple of things about aldolase. One, that it's tetrameric, and we can see that very, very easily because helpfully this diagram is in multiple colors, but we can see 1, 2, 3, 4 portions of aldolase. And if you forgot from previous slides, the function of aldolase is to cleave that fructose 1,6 biphosphate into DHAP and G3P. And yes, that is a reversible reaction. So here is a zoomed in representation of that particular step in glycolysis. There are multiple types of aldolase that exist. There's 2 main classes though. Type 1 aldolases, which are going to be found in plants and animals. So we have type 1 aldolases. And there's type 2, which exist and are found in bacteria and fungi as well. But aldolase 1, the one that we're going to discuss, has some isoenzymes. We have aldolase A, B, and C. Aldolase A has been isolated from muscle tissue, B has been isolated from liver tissue, and C has been isolated from brain tissue. But that's not to say that it doesn't exist anywhere else in the body, because all 3 of these do have multiple points where they could be expressed. In this diagram over here, and this is a diagram looking at expression of aldolase A, B, and C in frogs specifically. So we can see some expression of the different isoforms in the stomach, the intestine, liver, lung, kidney, brain, heart, muscle, skin, testis, and ovaries. You'll notice that muscle, by and large, is only going to express aldolase A, which makes sense. If it was isolated from muscle tissue, that would be the most popular isoform expressed there. Aldolase B is going to be popularly in the liver and the kidneys. And then C, again, it's a little hard to see because this diagram is black and white, but is going to be expressed in the brain and also the heart a bit. So to discuss how aldolase and glycolysis relate to one another, as we've already seen, aldolase is a glycolytic enzyme that's going to cleave that fructose into 2 3-carbon sugars, that DHAP and that G3P, in a reversible step. Now, mostly aldolase A's and aldolase C's are going to function in glycolysis, which, if you think about it, makes sense, because aldolase A, as I had stated previously, is found mostly in muscle cells. Now, muscle cells are going to be very metabolically active. They're going to have to produce a lot of energy, and glycolysis being that first step in cellular respiration, and that's part of producing energy. So it would make sense that aldolase A's are mostly to do with glycolysis and not gluconeogenesis. But for gluconeogenesis, you're going to see a lot of aldolase B and the liver being involved with that, and that makes sense too, because if we think of gluconeogenesis as glycolysis in reverse, producing glucose molecules, not for the purposes of direct production of energy, but to create a chemical form of energy that can then be used for cellular respiration. The liver is involved with maintaining maintaining good blood balance, maintaining a lot of things. So aldolase B being involved with gluconeogenesis makes sense. Discussing the aldolase B isoform a little bit more, the gene that codes for aldolase B is 14,500 base pairs long, approximately 364 amino acid polypeptide. And once again, you're going to see that tetrameric structure, which I had mentioned before in the other diagram. We can see it here, subunit A, B, C, and D. If you have mutations in aldolase B, it can actually result in hereditary fructose intolerance, the inability to process fructose. And those mutations, the reason that that happens is because those mutations are going to affect substrate affinity and enzyme stability. So if that enzyme that functions in that particular glycolysis or gluconeogenesis step cannot do its job, cannot bind onto that fructose molecule and cleave it, or do the opposite, then your body can't do anything with fructose. And so looking at that diagram, any mutation spots are going to be represented by these dots over here that I'm circling right now. So to move on and talk about the expression and regulation of aldolase B, that gene for aldolase B is going to be expressed in hepatocytes, which are liver cells, kidney cells, and enterocytes as well. So already mentioned how aldolase B is expressed in liver cells anyway, but here's those other places are places where you can see that expression going on as well. It's stimulated, the aldolase B gene is stimulated by glucose and insulin, and it's going to be inhibited by cyclic AMP. But the expression itself can be regulated by several different transcription factors. We have HNF1, HNF3, CEBP, and DBP. So HNF1, hepatocyte nuclear factor 1, is going to activate the expression of aldolase B, whereas HNF3 is going to decrease the expression of aldolase B. CEBP is an enhancer binding protein that is part of the zipper, the basic zipper or B-zip family of proteins, and that particular thing is going to interact with the CCAT box motif, which will effectively induce transcription. And it does that by interacting with the basal transcription apparatus. And then finally, we have DBP, which is D-site of albumin promoter binding protein. And that's going to bind to a promoter of the insulin gene and activate the expression of aldolase B as well. So looking over to the left, we're going to see an example of the HNF, the hepatocyte nuclear factors, in the purple and green diagram. And then we're going to see the CEBP down here on the bottom left, and finally the DBP there in the middle there. So to talk a little bit about why each of these activates or inactivates or anything like that, aldolase expression, these aren't going to directly regulate the expression of aldolase, but aldolase itself plays a hand in glycolysis and gluconeogenesis. So these factors are going to regulate things that also have a hand in glycolysis or gluconeogenesis. And in so doing, like let's say with HNF1, HNF3, if those stimulate glycolysis or decrease it from happening, then that will also affect aldolase expression, since aldolase is part of that process. So after that, again, if I were to propose this or present this to students, I would want them to walk away with something to do after the presentation. So as a follow-up, I would assign my students to probably work in groups to create a poster or a presentation themselves about partially one of those transcription factors that we had just talked about. So I'd want them to pick one, and I would want them to describe the exact mechanism in which that specific transcription factor is going to act on a gene, and then a diagram showcasing that mechanism, as well as an identification of the specific part of DNA that that transcription factor is going to interact with. But I would want them also to talk about how that impacts aldolase, like how specifically that is related to aldolase. And then I would want them to identify a specific protein other than aldolase that might be affected by the activation of that transcription factor, and then describe how that could in fact affect aldolase as well. So thank you for listening. And here are all the references I used in this presentation. Have a great day.